Good morning, I guess. It's evening where I am, but good morning. BMB510 MNE525 class. This is the first of the two online lectures for this week on biochemical kinetics and cellular metabolism. So sorry that this is about a day late. I had definitely meant to put it up yesterday, but I've been kind of busy here, so it'll have to do today. And greetings, I guess, from Krasnoyarsk, Russia, which is where I am right now. About 11-hour time difference here. I'm still fairly jet-lagged also, but I am here attending a conference. And hopefully all of you are enjoying your week. Okay, first of all, I'm going to start with a warning. A big warning sign. Your biomolecules are unstable. You should know this. Your important biological substances that are known to be unstable in you include DNA, RNA, proteins, polysaccharides, all the big ones. So do we panic? Well, not really. What is meant by unstable? Well, it is true, it certainly is true, important biological macromolecules are thermodynamically unstable. And I mean unstable with respect to hydrolysis. And that includes the list I mentioned: DNA, protein, RNA, polysaccharide. These can easily be broken down into their component monomers, whether we're talking about nucleotides for DNA, amino acids for protein, sugars for polysaccharides. They can be broken down by interaction with water. How is this not happening to us right now? Well, it should go without saying that this is not a rapid process that happens. The hydrolysis of these molecules is not fast. If it was fast, we would really struggle to stay alive. But in most cases, hydrolysis of our macromolecules is not going to happen while we're just sitting down at the breakfast table. So that lets us enter into talking about reaction rates. So we are basically made up, we're big bags of cells and enzymes. Enzymes basically are proteins in our body that do important things for us, that convert one compound to another, and they do all sorts of useful functions that allow us to exist. And the study of kinetics is the study of how fast reactions and other processes like this occur. And we typically study kinetics by first talking about reaction rate, which is typically expressed, if we're talking about enzymes, in the change of a concentration of a substrate or a product per unit time, or dc/dt, as we see right here. For many chemical reactions, though, it does not matter on which substance one bases the rate. So like I said, it could be change in concentration of substrate, or it could be change in concentration of product over time. Since the rates at which chemical species are consumed or produced, these rates are stoichiometrically related to one another based on whatever the stoichiometry of the specific reaction that we want to study is. So for example, we look at a fake reaction, A plus B yields C. The rates would look like this. We have dc/dt, the change in concentration of the product C, is equal to the negative change of the reactant A over the course of time, which is also equal to the negative change in the concentration of B, another reactant, over time. And this seems pretty simple, right? But this is not always the case. If stoichiometry equation that is used hides an intermediate step in the process, then the rates may well not be directly related to one another in the simple way that I just talked about here. So let's take a look at that. If the end result of our chemical process happens to be multi-step. So we've got A plus B yields some intermediate pair X and Y, and then X and Y eventually become C. The result, certainly you can argue, is that A and B are reacting, and then C is being produced. That goes without saying. But what we're not saying, it's kind of the sin of omission, is that the actual process is a two-step process, and first we're making this X and Y, which is then being converted to C. And also note that at the end of the reaction there may well be no X or Y present, as they exist transiently as intermediates. So the rates for steps 1, X and Y being produced, and 2, X and Y being converted to C, may be widely different. And so if we have these unrelated rates, we have, say, A and B reacting very quickly to form X and Y, and then X and Y reacting very slowly to form C. And then what we will see is that the change in the concentration of C over the change in time is not equal to the negative change in the concentration of A over the change in time, rather. This is since dA dt is very large in magnitude and dC dt is ultimately very small. All this aside, we can still write as our chemical equation A plus B yields C, but just know that this does not represent the rates of this. The rates won't be related to one another in the way that we showed from the very beginning there. So for chemical reactions that are single step, one can use the stoichiometric equation to develop reactions between rates. So if we have A plus 2B yields C, which would lead to the rate equation V is equal to change in concentration of C over time equals the negative change in the concentration of A over time equals negative 1/2 the change in concentration of B over time. Or to put it more general, if we have a reaction of the form AA plus BB yields CC, where small a, small b, and small c are our coefficients, this leads to a rate equation that looks something like this. We have 1 over c dc dt is equal to negative 1 over a dA dt equals negative 1 over b dB dt, etc. Many reaction rates can be modeled accurately using the following rate law, where we have the rate dC dt is equal to negative k times the concentration of A to the exponent alpha times the concentration of B to the exponent beta, where k is our reaction rate constant. If either A or B is absent, then dC dt is going to be 0. And in general, the more A and B you have, the faster C will be produced. Where alpha and beta are our constants, our exponents here, are greater than 0. And the total order of the reaction, N, is equal to alpha plus beta. So let's think about certain reactions. Some reactions are very energetically favorable. Molecules will react every time they collide with each other. For example, the coming together of a proton and a hydroxide ion to make water. So this will react very readily if these molecules are to collide together. Molecules in solution diffuse through the solution and run into each other as a result of diffusion. So this we know. The reactions that depend entirely upon the rate at which molecules collide, therefore, are called diffusion-limited reactions. But sometimes reactions don't always occur when molecules collide. Sometimes they react, sometimes not. These are known as reaction-limited. And there are two fundamental obstacles that can prevent colliding molecules from actually reacting with each other. One is an energetic barrier, and one is an entropic barrier. So you could have reactants moving too slowly, not enough energy as they meet. Reactants aren't facing the same way. That might be more of an entropic barrier. Reactants are energetic and oriented correctly, giving us the chemical reaction. So we've overcome the energetic and the entropic barrier to get the molecules to react. And as you might imagine, Gibbs free energy isn't too far away here when we're talking about chemical reactions. So if we can consider our old A plus B yields C equation, we can think of in terms of what is happening to the Gibbs free energy as the reaction occurs. So suppose delta G is negative, so that molecules A and B will decrease their Gibbs free energy by forming C. Now, does that necessarily mean that any two molecules of A and B will react? Well, if the Gibbs free energy of the reaction process is like that of the left graph here, then yes. So they'll go to a lower Gibbs free energy state. But if the Gibbs free energy of the reaction process is more like this on the right here, where we have a higher Gibbs free energy of the intermediates, then the reaction may well not occur. And these are transition states that we're talking about here. So when I say the reaction may not occur when molecules collide, it is because molecules must make it over basically a hill of energy, known as activation energy, at an intermediate step, such as a transition state. Whether two molecules, when they do meet, have enough energy to make it over the hill, this will determine whether they react or not. Statistically, molecules collide with a distribution of energies. The reaction moves from left to right often. It moves in the reverse direction very seldom. So the equilibrium constant and the reaction rate constants are closely related. So again, we have A plus B yields C. If we have a reaction rate constant for the forward reaction, this is K1, and a reaction rate constant K negative 1 for the reverse reaction, equilibrium will be reached when the amount of C being created is equal to the amount that's being converted back into A plus B. So for this reaction, the forward reaction rate dictates the rate at which C is being produced. So dC dt forward is equal to K1 times A times B. The reverse reaction dictates the consumption of C. So we would have K negative 1 is times C, the concentration of C, is equal to the reverse dC dt. Therefore, we can put this together. K1 AB minus K negative 1 should be negative 1 there. C is equal to 0. So again, this should be negative 1 here. If we rearrange this equation, we get K1 over K negative 1 is equal to concentration of C divided by concentration of A divided by times concentration of B. As we can see, the right-hand side of the equation is the definition of an equilibrium constant. So for this reaction, this A plus B yields C, the equilibrium constant is K1 divided by K to the K minus 1. Okay, so we consider reaction rates. We will consider rate limiting steps. And you might be familiar with this concept from general chemistry. So as the slowest step in the process, you have your rate limiting step, or what is also sometimes known as the rate controlling step. So metabolic networks in cells generally involve a large number of sequential reactions. This is true of most metabolic networks, including especially glycolysis and other important metabolic pathways. And products of one reaction are the reactants of a subsequent reaction. Biochemists have identified several of these rate controlling steps in several different networks. So here's an example. We have glycolysis, the glycolysis pathway. The first reaction, the phosphorylation of glucose, where glucose plus ATP yields glucose 6-phosphate plus ADP plus hydrogen, or protons, hydrogen ion. The delta G is very favorable, negative 18.8 kilojoules per mole. The equilibrium constant is defined as follows. K is equal to the concentration of ADP times the concentration of glucose 6P divided by the concentration of glucose times the concentration of ATP. And that K is equal to 2 times 10 to the third. Steady state metabolite concentrations in liver have been measured of these metabolites. Plugging those values into the above equation yields a ratio of only 0.02. Thus, this indicates that the reaction here we're looking at is far from equilibrium, that 2 times 10 to the third. It's quite a ways off. So if the steady state concentration ratios of these compounds is 0.02, how do we know the reaction is far from equilibrium in the liver? And the concentration of products relative to reactants is much lower than that what would be achieved at equilibrium. So that is one reason how we know. Consider the next reaction step. Glucose 6-phosphate yields fructose 6-phosphate. And the delta G for that is 2.5 kilojoules per mole. Our equilibrium constant for this reaction is K equals 0.36. Our physiological ratio is 0.31. So not close to the difference that we see in glucose kinase, suggesting that the reaction is close to equilibrium. And this reaction is not likely to be a rate determining step. Sometimes reaction steps that aren't rate limiting normally could indeed become so under certain, sometimes troublesome conditions. So look at the lactate dehydrogenase reaction, where the lactate plus NAD is yielding pyruvate plus NADH plus a hydrogen ion. Generally, this reaction is considered to be at equilibrium, and when NAD is depleted, this isn't the case. So when does that happen? When NAD is depleted during alcohol consumption. So if you go out to a bar and have a lot of some kind of drink, you are depleting your NAD. And you can see the reaction here for alcohol dehydrogenase. And the depletion of NAD can impair the lactate to pyruvate conversion. And they can impair it to the point where the individual can actually become hypoglycemic, even if they may not truly be in that state. The lack of NAD because of alcohol consumption can make you think so. It can make it seem so. But what all this does is suggest that the reaction is rate limiting under the conditions of alcohol consumption. So rate determining steps often will correspond to one of two things. Either the enzyme subject to the regulation by the environment, or we have enzymes that are irreversible. They can't go backwards. This is the case for the glucose conversion to glucose 6-phosphate. Because of the large negative delta G of the reaction, it is irreversible. And also because the glucokinase is subject to feedback regulation by its product, suggesting that it is indeed subject to regulation by the environment. There are other steps that aren't at equilibrium in glycolysis, suggesting that these steps are not, they are, as a matter of fact, rate limiting. They're rate determining steps. In fact, the overall rate control in glycolysis cannot be ascribed to a single step for this reason. So let's look at some of these here. Here is a picture of the pathway of glycolysis. So this is under typical physiological conditions. And we see, of course, we talked about already the hexokinase reaction. It's irreversible. It is controlled by the environment. It is a rate determining step under normal physiological conditions. Same with the phosphofructokinase, it is irreversible. Same with the pyruvate kinase here, which is also irreversible, converting phosphoenolpyruvate to pyruvate. And we can also look at not just kinetics of biological and biochemical reactions, but we can look at kinetics of drug binding to a target, to a drug target. So if we look at the drug proflavine bending to DNA to stop, binding to DNA rather, to stop cell division. And so the reaction equation is as follows. We have P plus D yields PD. So proflavine plus DNA yields a proflavine DNA complex. After initiation of this reaction, it is found that negative PDT is not equal to dPD dt. And this suggests that there is an accumulation of an intermediate, some kind of intermediate here in this drug binding to DNA, i.e. this tells us that the reaction is not simply a single step. So dP/dt is an extremely large number. It's a large negative number. This suggests that the P plus D reaction is diffusion limited. So it leads to an intermediate product. P plus D yields PD star or some such thing, yields PD. And PD star conversion to PD is indeed reaction limited. It is known further that PD star yielding PD is a unimolecular process involving rearrangement of a molecule. How do we know this? It would be pointless to aid or inhibit the P plus D step, since the PD star yields PD is the effective rate limiting step. Okay, let's look at some orders of reactions here. And of course, let's start at zero order reactions. So again, we have AA plus BB plus C yields C, where dC dt is equal to concentration of A to the power of the coefficient A, and the concentration of B to the power of the coefficient B. So a zero order reaction means that dC dt is constant and does not depend on A or B, the concentrations of these two molecules. One good example for conversion of ethanol to acetaldehyde by the liver enzyme LADH comes to mind. And the oxidizing agent in this case is NADH, and the LADH, the lactate dehydrogenase, does not change as a result of this reaction. So we get a rate here is equal to the disappearance of ethanol over time, or negative D concentration of ethanol divided by dt is equal to the change in the concentration of acetaldehyde over time, which is equal to the K0. It's simply equal to that reaction constant. First order reactions are slightly different. Say we have A to B, we have a unimolecular reaction. dB dt is equal to Ka, where K is the reaction rate constant. For a single step reaction, dB dt is equal to the negative dA dt, so the change in the concentration of B over time is equal to the negative change in concentration of A over time. Therefore, dA dt, or negative dA dt, is equal to the constant, the reaction rate constant times A. And we can integrate this to get the equation right here, where the natural log of A minus the natural log of the A0 is equal to negative KT. And if we rearrange that, we have the concentration of A equals the initial concentration of A times e to the negative KT. Okay, now let's consider time constants. there are two time constants commonly identified in chemical reactions. The time it takes for the concentration to reach the value of A0 divided by E. Also the half-life of the substance, the T1 half. And you can see the calculation of T1 half right here, and it's quite easy. Okay, so we've heard half-life before. Probably a lot of you have heard it from me quite a bit. So what is one application for the half-life? And it's a common use that's still used today, the common use of half-life in radioactive decay and carbon dating. So carbon in the biosphere, that's where we live, contains a known and fixed percentage of carbon-14, which is a kind of mildly radioactive form of carbon, and that decays in a single step first-order reaction to carbon-12. So carbon-14 will decay to carbon-12 in the prototypical single-step first-order reaction here. The creation of carbon-14 is caused by bombarding of carbon molecules high in the atmosphere by solar radiation. Okay. So as long as an animal or plant is alive, therefore, it's constantly going to absorb new carbon, which has a fixed percentage of carbon-14. There's a fixed percentage of carbon-14 in the atmosphere that we are taking in, or in the biosphere that we are taking in. Since that percentage is fixed, and indeed can be known at the time of death, one measure for this is the carbon-14 present in the fossil remains, and since we know the half-life of carbon-14, we can calculate exactly when a plant or animal died here, like this woolly mammoth. We can figure out how much carbon-14 they have remaining. If they have 100%, they weren't born too long ago, or they didn't die too long ago. If they have 6%, let's say, maybe they died over 22,000 years ago. And so this process is known as carbon dating. And it works because the carbon-14 decay is a first-order reaction with a known reaction rate constant. The clearance of small molecular weight drugs from the bloodstream also generally follows the first-order connections. For this reason, the half-life of the drug is often used to describe how long the drug remains in your circulation. Alright, now let's move to second-order reactions, or everything you ever wanted to know about denaturation and renaturation of DNA, but were afraid to ask. And so I say that because renaturation of DNA is a second-order reaction. We may be able to understand this better if we consider renaturation happening gene by gene. So in order for DNA to renature, gene A must recombine with its complements, in this case B. So A plus B is going to yield R, where R is our renatured double-stranded DNA. And so we can do a rate constant equation for this. We have dr dt is equal to K times A times B. And when we think of it, this is pretty obvious that the concentration of gene complements, B, is exactly equal to the concentration of gene A, right? Of course, because DNA is double-stranded, and not too long ago, gene complement B was actually bonded, hydrogen bonded most likely, to gene A. So we can change that rate equation a little bit and say dr dt is equal to K times concentration of A squared. And that makes renaturation a second-order reaction. The reaction as a single step might look something like this. dr dt is equal to negative dA dt, and dA dt is equal to negative K times the concentration of A squared. Or if we integrate that, we get 1 over A equals 1 over A0 plus KT. Okay, so by the way the renaturation reaction proceeds, we can learn about the complexity of specific DNA. So for every gene A, there may or may not be a redundant copy somewhere else on this DNA molecule, where N is equal to the number of base pairs between gene A and its redundant copy. So this may or may not happen, but we have a way to calculate it anyway. So if A is unique, then N is just going to equal the entire length of the DNA molecule we're studying. However, if N is equal to 1, then the DNA is simply an artificial replication of gene A over and over and over. So if we denote C0 as the concentration of all base pairs before renaturation, then the concentration of base pairs in type A can be calculated like so, where A0, the concentration of A0 is equal to C0 divided by N. If N is equal to 1, the entire concentration of DNA is all type A. As renaturation occurs, we can further define terms such as F, that is the fraction of molecules that are reassociated. So F is equal to the concentration of A divided by the initial concentration of A. So there's that F equation again, and we can consider our previous equation for the renaturation reaction that we solved for by integration. Our 1 over the concentration of A is equal to the 1 over concentration of A0 plus KT. So we can plug the first equation into the second here. We can plug this equation into this and solve for F. And we get F equal to 1 divided by 1 plus A0 over T, KT, the concentration of A0 multiplied by KT here. And then we want to write the F equation in terms of N. And if you recall what N is the number of base pairs between gene A, whatever gene A is, and its redundant copy. So if we put our F equation in terms of N, we end up with F equals 1 divided by 1 plus initial concentration multiplied by KT divided by N. And so we can begin to visualize some of this stuff by looking at DNA reassociation curves. We can look at the percentage of remaining single molecule or single-stranded DNA over the course of which is a product of the concentration at the time. So for example, if we had a run of poly U and a run of poly A in our DNA, then that would come together much quicker than, say, if we have calf non-repetitive fraction DNA or E. coli DNA or Ralstonea eutrophic DNA for that matter. It takes longer to reassociate the more complex the DNA molecule. So in this case, the fraction of the reassociated DNA has a function of C0 times T. DNA renaturation data are also presented in terms of half-life. We can calculate when pairs have half combined. So we can calculate this at the half-life here. So half of combined, half of non. And T equals N divided by the concentration of DNA at time 0 multiplied by K, the constant. We can also use that, rearrange it to calculate K, where we get K is equal to N divided by concentration of molecules times T1 half. And for poly A and poly U, and if you're wondering, those of you that aren't finding yourselves too biologically savvy, if you're wondering what poly A and poly U might look like, here is a schematic showing a base pair of the adenine to the uracil. and how that can happen, how they can form double-stranded molecules. So if we have a double-stranded molecule or two single-strands molecule, one's poly A, the other's poly U, we can calculate our K very easily because N is equal to 1. So that makes this reaction here a lot simpler. We can use a parameter to quantify complexity of DNA, and that parameter is known as kinetic complexity, or XC, where XC is equal to the N total divided by the N. And little n here is the number of repeated sequences in the original DNA, and N total is the total number of genes. And it should be known that, in fact, maybe it even should be obvious that XC, the complexity here, is equal to N. And what is N again? We'll go back to it one more time, where N is the number of base pairs between the gene A and its redundant copy, if it indeed has one. And so, same here, that's the kinetic complexity, XC or N, as we know them. Okay, let's think a little bit about the mechanism of DNA reassociation. So this is the actual mechanism by which DNA is believed to renature. And it consists of two steps. Our first step is the complementary fragments joining together and binding, although their binding is very weak, making this like two base pair bond here. This bond, because of the size of the entire DNA chain, this bond is usually unstable as we see it, and the fragments will end up dissociating. So they'll go, basically they'll associate, dissociate, associate, dissociate, like that. But sometimes the fragments join together in such a way that an arrangement of a third base pair is able to form. And in this case, the reaction is much more stable, and renaturing the DNA continues all the way to completion. So once we have that third base pair, we go all the way to our product of fully renatured DNA. And so again, the reaction consists of one quick process and one rate-limiting process. And that rate-limiting process, as you may have guessed, is the second step. So step one is A plus B, our unstable bond here, the making of our unstable bond, as well as the breaking of it. This is fast. And step two, the unstable bond forming the product, which is our stable DNA duplex molecule. And the rate of renaturation is going to be governed by the rate of step two here. So the tentatively bound DNA molecule to the more stably bonded DNA molecule with 3 bonds here, such that the change in the amount of product is equal over change in time is equal to K2. Since reaction 1 is so fast compared to reaction 2, the amount of the unstable helix is governed by a step 1 equilibrium condition. So we have our equilibrium equation is unstable helix divided by the concentrations of A and B, or it could be rearranged to form the concentration of unstable helix is equal to K1 times A times B. And so we can combine with we can combine with the dp dt equation with K2 multiplied by the concentration of unstable helix to give us this new equation here. The dp dt is equal to small k2 times big K1 multiplied by concentration of A multiplied by concentration of B. Okay, so we have that reaction equation. Let's also consider the overall reaction equation that we put together here. We combine the two reaction equations in order to get big K is equal to little k2 times big K1. And so we can get our K from the overall reaction. K is equal to 10 to the 6th meters per meter per second. And the equilibrium constant for step 1 is found to be K1 equals 10 to the negative 1 per mole. So we have 2 of the 3 values here. We can easily calculate the third as K2 is equal to 10 to the 7th per second. This is an important rate because the formation of stable helixes is essentially the basis for genetic information transfer in biological systems here. Okay, so I'll let you in on a little secret. That's all the slides I've made. So that's all the talk I can give for right now. I will continue to build the slide deck in order to finish this section here, to finish our week of biochemical kinetics. I'll post this, and on Thursday I will post a second lecture, I will post the slides, and I will post a little assignment. So until then, have a good week, and I will talk to you shortly.